Okay, so I have the first lecture today, and I'm talking about signal transduction. And basically, the bottom line is that low doses of oxygen as well as NO that will stimulate proliferation of cancer cells, whereas if you have very high doses, it can cause apoptosis of the cancer cells. So what happens in the central nervous system as well as other cells is if you elevate the cytosolic calcium, and in particular if you open calcium channels, then that will activate the nitric oxide synthase, and the nitric oxide synthase then uses arginine as a substrate to make nitric oxide and citrulline as products. And then the soluble guanylyl cyclase uses GTP as a substrate, and the nitric oxide then activates the guanylate cyclase to produce cyclic GMP, which functions as a second messenger in the cells. So here's a little cartoon. And so we see then that the calcium goes through calcium channels to elevate the cytosolic calcium. And then low levels of the calcium will activate the nitric oxide synthase to produce nitric oxide. And the guanylate cyclase here, it's shown in green, and the star indicates that it's got an iron in it. And the nitric oxide then binds to the iron, causing a conformation change in the enzyme so that GTP gets metabolized then to cyclic GMP. And also in the plasma membrane, we have what's called the atrial natriuretic peptide receptor. It's abbreviated ANP, and it's got two subunits then. And so these outside subunits bind the ANP, and then it causes a conformation change. There's a kinase, and then there's a guanylyl cyclase component to these proteins. And the A and the B form a dimer, and then that activates the guanylyl cyclase to produce the cyclic GMP. And then the cyclic GMP, it's a transient elevation. It occurs over a several-minute period. And then the cyclic GMP is degraded by phosphodiesterase. So here's the ANP receptor then. We see the A and the B chain. They're large proteins, over 1,000 amino acids. The molecular weight is a little over 100,000. And there's a signal sequence then, and an extracellular sequence that binds the ANP. It crosses the membrane just once, and then we have on the intracellular side, there's a kinase activity, and then there's a guanylyl cyclase activity. And we see that the guanylyl cyclase, it's just about 130 amino acids long. And here's the soluble guanylyl cyclase. And we have two subunits, an alpha-2 and a beta-1. They're both over 600 amino acids. Molecular weights 70 to 80,000. And the guanylyl cyclase component of this, again, it's about 130 amino acids. So the difference is the ANP receptor binds AMP to elevate the cyclic GMP. The soluble guanylyl cyclase, it responds to calcium. And then the nitric oxide synthase makes nitric oxide, which then activates the guanylyl cyclase. So in particular, the nitric oxide binds to the iron that's present in the heme domain. And so then this stimulates the dimerization, and the catalytic activity then gets turned on. So the soluble guanylyl cyclase, it's got three domains: the heme binding domain, the dimerization domain, and the catalytic domain. And then the cyclic GMP has 4 protein targets. One of the targets is protein kinase G. So this is a protein then which will phosphorylate serine and threonine amino acids on various protein substrates. And this ultimately leads to vasodilation. The protein kinase G2 phosphorylates the cystic fibrosis conductance regulator, but we're more interested in the protein kinase G1, because this is leading then to the vasodilation that the nitric oxide initiates. Also, cyclic nucleotide gated channels are present in neurons, leading to visual signals, leading to nerve impulses. And then we mentioned before that the phosphodiesterase reverses the activity of the guanylyl cyclase. It metabolizes the cyclic GMP. But then Viagra, it selectively inhibits the phosphodiesterase 5. So as a result, then, when you have nitric oxide, it stimulates the guanylyl cyclase, leading to the vasodilation. And this vasodilation is retained if you inhibit the phosphodiesterase. So in the laboratory, then, we can elevate the nitric oxide using the nitric oxide delivering agents. And one of these is Spurno. And it was shown that low concentrations of Spurno then increase the cyclic GMP after 30 minutes. So the nitric oxide then, it's activating the nitric oxide synthase, and then the guanylyl cyclase is activated, leading to the increases in cyclic GMP. And also the P44, P42 MAP kinase, which we call ERK, it gets tyrosine phosphorylated. And this then leads to a proliferative signal. The phospho-ERK goes into the nucleus and stimulates AP1 sites on various genes. So when ERK gets elevated, then you'll see increases in C-phos and C-Jun in the nucleus. And then these can form heterodimers, activating AP1 sites, turning on genes. And one of the genes that gets turned on is the VEGF gene, which Dave Roberts talked about last week. So it then leads to an angiogenic response. So in summary then, we see that low doses of NO will cause an elevation of cyclic GMP. So the nitric oxide synthase gets activated, then the soluble guanylyl cyclase gets activated, elevating the cyclic GMP. And the cyclic GMP can lead to phosphorylation of various proteins through the protein kinase G. One such protein that gets phosphorylated is the ERK, and especially when the ERK gets tyrosine phosphorylated, this leads to a proliferative signal. But high doses of NO will cause apoptosis of the cancer cells. And in particular, the high doses of NO can induce stress proteins, disrupt mitochondria, release cytochrome C, and activate caspases. So we can do all these experiments using cancer cells in the lab. We can add Spurno to our lung cancer cells, and we see then that the P53 gets phosphorylated, and this then leads to less G1 to S transitions in the cell cycle, and in turn, ultimately, the P53 can get inactivated, leading to increased apoptosis of the cancer cells. So also in the lab, we can use nitric oxide donors, such as S-nitroso-N-acetyl-penicillamine, abbreviated SNAP, and sodium nitroprusside, abbreviated SNP. These chemicals were especially popular in the 90s and the 2000s. You use the nitric oxide delivery agents. But ultimately, this causes apoptosis of lung cancer cells. So here's an experiment that we did where we're adding SNAP, various doses, to the lung cancer cells. And we see that if you're using millimolar doses, the growth goes down. And in turn, the nitrite concentration is elevated. And it's possible that the nitrite can be metabolized to the nitric oxide as well. Here, we've added the nitric oxide delivery agents, Dino or Papano, and we see that the growth goes down in the lung cancer cells. So here you can measure DAF reactive chemicals within the cells, and after you add Papano, you see that the fluorescence increases within minutes. So the nitric oxide is then released from the delivery agents into the cells, and this contributes to a response in the cells. And here we've done a colony forming assay, and we've added Papano, and we see when we add 100 micromolar concentrations, the number of colonies goes down, 2 different lung cancer cell lines. And another thing that you can do in this colony assay is you can just add macrophages to it, because they don't really interact with the cancer cells and they don't form colonies, but what they do is they can deliver the nitric oxide then. And you see the colony number goes down as you add more macrophages to your cultures. So we saw before the P4442 MAP kinase was important in proliferation, but here, if you add high doses of nitric oxide, you activate the P38 MAP kinase, which is a mediator of caspase 3. And caspase 3 then leads to apoptosis. So using the MAP kinase inhibitor SB202-190, this protects cells from nitric oxide-mediated cell death. And SNP and SNAP decrease survival levels as well as BCL2. Survivin is critical for cell cycle progression. BCL2 is critical for cellular survival. So when these two go down, apoptosis goes up. So in summary then, high doses of nitric oxide can decrease BCL2. It activates P38 MAP kinase, leading to cytochrome C activation. This in turn will then activate caspase 9 and then caspase 3, leading to cell death and apoptosis. So the point here about proliferation is low doses of nitric oxide can stimulate proliferation. High doses can inhibit. But physiologically, in patients, there's been no way to deliver the concentrations of nitric oxide that are needed to inhibit cancer cell growth. So this works fine in culture, but in vivo components are much more difficult to utilize. So we've looked then at the nitric oxide. We're going to look now at the oxidants. And we have 2 pathways again. Low doses of reactive oxygen species will cause proliferation, and in particular, the P4442 MAP kinase gets activated, leading to activation of AP1 sites, and that causes production of various growth factors that cause proliferation. But high doses of low doses of reactive oxygen species can also activate NF-κB, leading to inflammation and increased survival. of the cancer cells. So in terms of the reactive oxygen species, low doses again will go through the RAS pathway, activating MEK1, which stimulates ERK1 and 2, leading to growth. And then very high doses of reactive oxygen species will activate MEK3 and MEK4, leading to P38 activation, which we know inhibits growth. And also chunk can get activated, leading to various stress responses. So now we're going to take a look at what's going on in the cancer cells, because cancer cells don't work just by increasing reactive nitrogen species or reactive oxygen species. There's both species present in the cells at any one time. So one of the key things that we see here is the nitric oxide then reacts with the oxygen to generate nitrogen dioxide, which can react with nitric oxide to yield dinitrogen trioxide. This can be metabolized with water to nitrate, or we can then target nitrosylation of key proteins. And so here we see in a cell, we have nitric oxide being produced by nitric oxide synthase. So we have nitric oxide forming from NOS. Arginine is the substrate as well as O2. And then the nitric oxide can go through the plasma membrane into the cell and do further reactions, generating nitrogen dioxide. And then we have the dinitrogen trioxide. And then we can nitrosylate various proteins. And so one of the proteins that can get nitrosylated is RAS. So this is a P21 GTPase. And what gets nitrosylated is a cysteine amino acid. So this has a sulfur then that can react with the nitric oxide. And when RAS gets activated, then it goes downstream, and MAP kinase, the P44, P42, gets turned on, leading to growth. And another thing that can get nitrosylated, as we'll see, is caspase 3. But when it gets nitrosylated, it leads to apoptosis. But if you denitrosylate the caspase 3, that is essential then for apoptosis. So nitrosylation of the caspase 3 inhibits its activity. When you denitrosylate, then that leads to apoptosis. And in the CNS, one thing that can really get nitrosylated is the NMDA receptor for glutamate, an excitatory transmitter. So in cells, high doses of hydrogen peroxide will kill them, but low doses can stimulate proliferation. And so we see then at low doses of ionomycin, which increases cytosolic calcium, NOS activity gets turned on. And then the cytosolic calcium goes up transiently when you add H2O2. Calcium channels open. When the channels open, then the calcium flows in. Nitric oxide synthase gets turned on. And then you can also increase calcium in the cells using ionomycin, which is an ionophore. So then the question is, how do the cells sense a cytoplasmic oxidative response? And of course, one of the key things initially is an increase in cytosolic calcium. And then that calcium we see will turn on various enzymes, such as nitric oxide synthase activity will increase. And we have nitric oxide. This can lead to nitrosylation. For caspases, nitrosylation will inhibit activity. Another thing that we're going to look at shortly is protein tyrosine phosphatases. They respond to both nitric oxide as well as reactive oxygen species. And then we've seen we have heme-binding proteins. Nitric oxide interacts with proteins that have iron, such as soluble guanylate cyclase. And then another factor that is rather new is the nitration of tyrosines. And this happens in certain proteins, such as P53 and NF-kappa B. So we're starting to appreciate then that the reactive oxygen species, the reactive nitrogen species, they're involved in many signal transduction pathways in the cells. We're discovering more and more all the time. And this just shows you the key step in terms of tyrosine phosphorylation is protein tyrosine kinases. We'll be looking at shortly. The EGF receptor causes phosphorylation of tyrosines. And this is basically a growth signal. And then this is a transient event, which occurs over a 5 to 10 minute period. But then the protein tyrosine phosphatases take the phosphate off, and you return back to the tyrosine, which is sort of the inactive state of the receptor tyrosine kinase. And we'll see that then reactive nitrogen species as well as hydrogen peroxide, it can oxidize key cysteine amino acids in the protein tyrosine phosphatase. So this is a pathway then that we've investigated in lung cancer cells. And so the nitric oxide will be stimulated by iNOS in the cancer cells. And then cyclooxygenase 2 will get turned on because it's got an iron at its active site. And when it gets activated by the nitric oxide, its activity is increased, leading to the production of prostaglandins. So then the prostaglandins, we'll see, can activate the EGF receptor, which is very much involved in growth, and it can cause angiogenesis through the production of VEGF. And this G protein coupled receptor, the EP2 receptor, is one of the key G protein coupled receptors in this process. So the cyclooxygenase then, we have phospholipids being metabolized by phospholipase A2 to arachidonic acid. And then the cyclooxygenase enzymes convert arachidonic acid into prostaglandins. The lipoxygenase enzymes can convert arachidonic acid into leukotrienes. So we're going to focus especially on the prostaglandins. And so the cyclooxygenase converts arachidonic acid into prostaglandin G2. Then the prostaglandin endoperoxide synthase converts this to PGH2, which then gets metabolized into PGE2, which we know is important in growth, PGI2, which is important in the CNS and neuronal sprouting, and thromboxane, which increases then the motility of various cells. So we're going to especially focus on the PGE2. So there's two types of cyclooxygenase enzymes in the cells, a COX-1, which is a constitutive housekeeping enzyme, and it's traditionally inhibited by nonsteroidal anti-inflammatory drugs. such as aspirin. But when you start giving aspirin to animals, you find there's toxic side effects. They can get stomach ulcers. And some people, such as myself, I can't take aspirin either, because that will cause trouble for my stomach. So then, in cancer, they found that certain cancer cells had high levels of COX-2. And this can be induced by various growth factors, such as EGF, transforming growth factor beta, tumor necrosis factor alpha, hypoxia, and UV light. And COX-2, it's inhibited by non-steroidal anti-inflammatory drugs, but then they started coming up with selective cyclooxygenase 2 inhibitors, such as celecoxib. So the cyclooxygenase 2, as well as the cyclooxygenase 1, they both have about 600 amino acids. And there's an iron binding site in each of them, and this iron can interact with nitric oxide. And there's a heme group in both cyclooxygenases. So a histidine interacts with the heme and anchors it to the protein. And then the aspirin can irreversibly acetylate a serine residue and inactivate the enzymes. But COX-2 is turned over very quick. It can be made very quickly, and it can be degraded very quickly. So we then did an animal model for lung cancer using the AJ mouse. You give the mouse carcinogens, and lung tumors result. And we looked then for COX-2 activity in the various lungs of the animals. And so here you see these nodules. These are adenomas, lesions that come from the carcinogen. And these adenomas develop 4 months after you give the animal the carcinogen. So this is really a prevention study. And so we look then for the COX immunoreactivity. We find it in the bronchus of the lung, which is the sort of the windpipe. And then we find it in adenoma cells, as well as bronchiola. And so here's the lung. And you see here lots of villi on the surface of the lung, and these villi are where the lung exchanges gases. You breathe in oxygen, you breathe out CO2. And then here's the bronchus, here with lots of cartilage. And we see then the adenoma as well as the bronchiola in the lung. So the COX-2 is sort of ubiquitous to the lung. It's present in many different places. And we used a non-steroidal anti-inflammatory drug, initially indomethacin in the 1990s, and it slowed the tumor growth. But if you didn't give any carcinogen, you didn't get any tumors in this animal model. So then celecoxib was developed, Celebrex. It's primarily used in arthritis patients. And in lung cancer patients, it was found to be a very weak drug. It potentiated some of the actions of the chemotherapeutics, but it's never sort of really made it big in terms of cancer treatment. It's much bigger in terms of treating patients with arthritis. So the celecoxib then in animal models, it reduced PGE2 levels by 80%, and it reduced endothelial cell proliferation as a result of reducing the VEGF in the cells. And in lung cancer patients treated with celecoxib and chemotherapeutic agents, Taxol and carboplatin, the serum VEGF declined. So in turn, there was less angiogenesis of the tumors. So then in the 2010s, we started developing additional drugs besides the celecoxib. And here we have a series of nitric oxide aspirin. You see is quite good at reducing PGE2 levels. And we got some compounds from Italy, S-valproate, L-diclofenac, S-sulindac. The valproate and diclofenac were very good at reducing PGE2 levels. And this is another COX-2 inhibitor, the DUP-697. It's very good at reducing PGE2 levels. So the PGE2 then, it turns out, can activate this G protein coupled receptor, the EP2 receptor. So this is a G protein coupled receptor, about 360 amino acids, and it crosses the plasma membranes 7 times, and it's a glycoprotein. So the amino acids on the extracellular surface of the receptor bind the prostaglandin E2, and this is just looking at messenger RNA. We see all the lung cancer cell lines have it. And the PGE2 binds with high affinity then to the lung cancer cells because they have the EP2 receptor. And you can inhibit binding with PGE2. PGE1's pretty good as well. And PGF2 alpha inhibits binding a little bit, but PGG2, PGI2, PGD2 are pretty much ineffective. And then it turns out we have one drug that's a G protein coupled receptor antagonist, this AH6809. So we started doing studies with it. And PGE2 is an agonist which increases the cyclic AMP in the lung cancer cells. And AH6809 is an antagonist which blocks the receptor. So if we add increasing amounts of PGE2, we'll see that the cyclic AMP goes up. But when we have the blocker, AH6809, it inhibits the ability of the PGE2 to elevate the cyclic AMP. In terms of the VEGF production, we mentioned before that nitric oxide donors will increase VEGF. And it turns out the VEGF is actually made in the cancer cells and secreted from the cancer cells. Then the VEGF diffuses to endothelial cells that have the VEGF receptors and stimulates the angiogenesis. So here we're looking at the VEGF mRNA, and PGE2 increases the VEGF mRNA, but so does EGF, which binds to the EGF receptor. And the PGE2, when it elevates cyclic A, it will activate protein kinase A, and H89 is an inhibitor of the protein kinase A. So we mentioned before the protein kinase G is important for nitric oxide. For cyclic AMP, it's the protein kinase A that causes the tyrosine phosphorylation and the growth. So in terms of colon cancer, if they knocked out the gene for COX-2, VEGF protein was reduced by 94%. And in non-small cell lung cancer patients, increasing COX-2 mRNA correlates with increased VEGF production, increased microvessel density, decreased patient survival, and early relapse from cancer. So COX-2 then is intimately involved in VEGF production and angiogenesis. So looking at the COX-2 then, we found that it could be induced by EGF. So we started looking then at what the EGF receptor was doing in these cancer cells. And so the EGF receptor, it's a traditional tyrosine kinase receptor. It's got two extracellular domains. Domains 1 and 3 are important in EGF binding. Domains 2 and 4 are enriched in cysteine amino acids. It crosses the membrane once, and when EGF binds, it causes a conformation change, increasing the tyrosine kinase activity. And then various protein substrates can get tyrosine phosphorylated. And you see we've got lots of tyrosine kinase receptors. In the neurons, you've got the NGF receptors. Most of the cancer cells also have IGF-1 receptors. And the VEGF receptor, important in angiogenesis, it's also a receptor tyrosine kinase. So the EGF receptor, it's big, over 1,100 amino acids. 621 amino acids are on the extracellular side of the plasma membrane, and they bind EGF. And then there's a 23 amino acid transmembrane domain. 542 amino acids are intracellular, and a lysine at 721 binds ATP and activates the tyrosine kinase activity so that you get tyrosine phosphorylation of various protein substrates leading to growth. But then a way in which the EGF receptor terminates its signal is it will tyrosine phosphorylate itself. And in the lab, we especially look at this tyrosine phosphorylation of 1068. So here's the EGF receptor. It actually forms a dimer when it binds the growth factor. And then it phosphorylates protein substrates, and one of these is PI3 kinase, and this then activates AKT, causing cellular survival. So the EGF receptor then will increase the survival of the cancer cells. BCL2 gets inhibited, and apoptosis gets inhibited. The EGF, it's a proliferative signal. So one thing that they found is that when you added EGF to cells having the receptor, it would actually increase hydrogen peroxide production in the cells. And we mentioned before that when the hydrogen peroxide goes up, the enzyme inactivating the tyrosine phosphorylation goes down. So P10 activity goes down. So when you add EGF, you get PI3 kinase forming. The PIP3 will activate AKT, leading to cellular proliferation. But the hydrogen peroxide probably gets produced by the NADPH oxidase. And a little later on in the course, Tom Leto will be giving a lecture on this particular NADPH oxidase. And then the peroxide causes reversible oxidation of the cysteines in P10. And this disulfide oxidation is reversed by the thioredoxin. So this is how cells work. We have many things going on at the same time. We mentioned, we have to worry about the reactive oxygen species and the reactive nitrogen species, and especially the nitric oxide. It can interact with irons in various enzymes, and then it can nitrosylate various proteins, especially through cysteines. So, a very curious thing is that when you add PGE2 to cells, it will cause the tyrosine phosphorylation of the EGF receptor. So this is telling us then that the G protein coupled receptor, the EP2 receptor, is somehow activating the EGF receptor. And this process is called transactivation. So we're not affecting the EGF receptor synthesis or levels in any way. We're just affecting its phosphorylation when we add the PGE2. So here we're adding the PGE2, getting the EGF receptor tyrosine phosphorylated. And an inhibitor of the EGF receptor is a tyrosine kinase inhibitor called gefitinib. So when we add gefitinib, we stop the tyrosine phosphorylation caused by PGE2 or caused by EGF directly. You see, you get a very big signal when you add EGF to lung cancer cells. Lots of tyrosine phosphorylation. PGE2, you get some tyrosine phosphorylation, but it's not as strong as when you're directly adding EGF. So in lung cancer then, they found that these tyrosine kinase inhibitors, they will actually can be used in patients that have EGF receptor mutations. It's very curious in that only in lung cancer are there lots of EGF receptor mutations. And in these patients with mutations, you can add gefitinib, and it will basically cause the cancer to stop growing. So there have been various clinical trials then, such as the IDEAL-1 and IDEAL-2 trial, and you add gefitinib to patients, and you try to cause a response in at least half of the patients. And so it turns out if the patient has mutated EGF receptor, they will respond to gefitinib. If they have wild-type receptor, it doesn't work very well in the patients at all. So the responsiveness then is not associated with EGF receptor levels, but rather with EGF receptor genetic mutations. And one of the ways they first figured this out is they did studies in the US, and they did studies in Asia, and they found the gefitinib was much better at working in the Asian population, especially Asian women who were non-smokers. They had lots of these EGF receptor mutations and responded to gefitinib. The Americans, we didn't have as many mutations, and so gefitinib wasn't as effective in our patients as it was in the Japanese. So then they did all sorts of genetic studies, and they found out that there were mutations in the tyrosine kinase domain of the EGF receptor. And especially if amino acid at 719 was mutated or 858 was mutated, these patients responded readily to the gefitinib. So now we've entered the age of molecular medicine in lung cancer. When patients come in, one of the first things they do is they do a genetic analysis of the tumors to see if they have the EGF receptor mutations. If they do, you put the patient then on the gefitinib. So look at how complicated this whole process is. We mentioned that the EGF, when you add it to the lung cancer cells, you'll increase COX-2 expression, and then that's going to increase prostaglandin levels, which is then going to interact with the EP2 receptor, the G protein coupled receptor. This is going to stimulate adenylyl cyclase, which is going to activate protein kinase A, which is going to cause phosphorylation of Krebs. This is going to go into the nucleus and then activate certain genes, such as the VEGF gene. So growth through the EGF receptor, through COX-2, ultimately leads to angiogenesis. So this is how big tumors then grow bigger, given more VEGF, so they stimulate more angiogenesis. But then we see that this is a cycle. That's what happens in cancer cells. They have these growth-promoting cycles that work in an autocrine manner. So when you have the prostaglandin, it activates the EP2 receptor. Then in turn, other proteins get activated, such as SRC, such as matrix metalloproteases, and then they cause EGF receptor ligands, such as TGF-alpha, to be released, activating the EGF receptor. And so we have a cycle then, a growth-promoting cycle, an autocrine growth cycle, working through prostaglandin E2. So this works very well in the small tumors, and then when the tumors get big, then we go down to this part of the process, increasing VEGF production, leading to increased angiogenesis, leading to growth of even bigger tumors. So this is what signal transduction is in cells. It's very complicated. There's many things going on. The cells don't respond just to nitric oxide. Reactive oxygen species are also involved. So this is an animal experiment then, where we're giving the cyclooxygenase 2 inhibitors, the S-valproate and S-diclofenac, respond best. The S-sulindac doesn't respond as well. We see we start out with very small tumors in the animals. Within weeks, we get to very large tumors. And at NIH, if you've ever done animal studies, you know you're not allowed to have the tumor kill the animals. When the tumors get to about 1,000, we have to euthanize then the animals. And here we see that the S-valproate at a dose of 18 mg per kilogram reduced the tumor size about 6-fold. And the S-sulindac wasn't nearly as effective. And these drugs didn't really affect the animal weights, so they weren't causing dramatic stomach ulcers. We were amazed when we sacrificed the animals and looked, and all the organs just looked normal. Everything seemed fine. It's just the tumors were a lot smaller in the animals. And so this shows that the nitroaspirin actually caused apoptosis in the animal cancer cells, whereas the S-valproate did not. So the nitric oxide aspirin was cytotoxic. The S-valproate was cytostatic. It was reversible. So if you had animals and you were giving it the S-valproate, and then you stopped administering it, the tumors would rapidly regrow. And a surprising thing that we found is these COX-2 inhibitors, they induced expression of E-cadherin. You see the E-cadherin increases dramatically with very low doses. You know, this is a nanomolar dose that's causing a dramatic response in E-cadherin. And when we looked at the tumors of the animals, we saw the same thing. There was an increase in E-cadherin, not as big as what we saw in the cells, but it was a significant increase. So there's one process in cancer called epithelial to mesenchymal transitions. And so the cells start out as epithelial, but as more and more things get mutated, the cells become mesenchymal, and then they're more resistant to traditional therapy, such as chemotherapy, and they're certainly more resistant to the effects of gefitinib. So these cadherins, they're cell surface glycoproteins that connect the cells to the extracellular environment. And here we see the receptor tyrosine kinase, the EGF receptor, and here's the E-cadherin. And it helps anchor the cells together, basically, the E protein. but we see there's lots of other proteins involved as well, including microtubules. So when we looked again, we saw the E-cadherin going up. Nothing was happening to the tubulin. Nothing was happening to the COX-2. Nothing was happening to the EGF receptor. Nothing was happening to the ERK. But we saw vimentin going down and ZEB1 going down. And this is just the process of the epithelial to mesenchymal transition. The cancer cells, they're initially localized to the tumor, but then they want to get out and invade to distant organs, and they become more mesenchymal to do this. So prostaglandin stimulates them to become more mesenchymal. In our COX-2 inhibitors, they basically were stopping the action of the PGE2. So other proteins involved are a protein called snail, and this protein ZEB1 is intimately involved, and when ZEB1 is turned on by PGE2, it inhibits E-cadherin production. And so the non-steroidal anti-inflammatory drugs that we were using were doing just the opposite. They were reducing ZEB1, they were reducing vimentin, and turning on E-cadherin. So they were making our cells go from the mesenchymal state back to the epithelial state, where they grow a lot slower. So another thing that happens is gefitinib and erlotinib are most effective in cancer cells that are of the epithelial nature. So the mesenchymal cells then, the gefitinib and erlotinib are not nearly as effective. So if you transfect then E-cadherin into lung cancer cells, you can actually increase the responsiveness of the cells to gefitinib. And so we then added the non-steroidal anti-inflammatory, the COX-2 inhibitors to the valproate, and we saw that the dose-response curve shifted, and the cells were then more responsive to the gefitinib. So this is sort of good in terms of therapy because most of the lung cancer patients actually have the wild-type EGF receptor, and they don't respond very well to gefitinib. And so if you add these non-steroidal anti-inflammatory drugs, the COX-2 selective inhibitors, you make the cells more responsive to the gefitinib, and this might translate into better therapy for the patients. So then in summary, we've seen that signal transduction can be very complex. There's lots of different things going on, and we've discussed at low doses of NO, the cyclic GMP increases, and that starts the whole process, such as the phosphorylation of the ERK, and then the P53 also gets phosphorylated. At low doses of nitric oxide, also we see the COX-2 is activated, leading to increased PGE2 and activation of G-protein-coupled receptors for prostaglandin E2. And the S-valproate and the S-diclofenac are cytostatic agents which inhibit COX-2, decrease the proliferation of lung cancer cells, and increase E-cadherin expression. So as a result of the increased epithelial content of the nature of the lung cancer cells, the S-valproate, S-diclofenac increase the potency of gefitinib in lung cancer cells which have wild-type EGF receptor. And so these are some of the colleagues that participated in these studies, especially our good friend, David Wink. We always want to acknowledge him. So that's the end of my lecture. Are there any questions? Yes? Well, to do that, you have to do additional animal studies, and you have to show that the compounds have very reduced toxicity. But in general, at NCI, what we're always doing is trying to translate our studies in the laboratory to the clinic. And so about 30% of the drugs that are used in cancer patients were developed here at NCI. We have a very good relationship with the FDA so that we can get our compounds into clinical trials. And here in Bethesda, we especially do the phase 1 and phase 2 trials. Then the phase 3 trials are done at many different institutions all around the US. And to do that, we have to get a pharmaceutical company participating who can supply the drugs for all the clinical trials. So we're very much oriented in the bed-to-bedside type of research. Okay, we're going to have Pal talk now. And Pal's at NIAAA. And he got his MD degree from Semmelweis University in Hungary, and subsequently he got a PhD and came to the US, and he was at Thomas Jefferson University in Philadelphia for a couple years, and then he came to NIH, and initially he did research in the lab, but now he's a tenured principal investigator, and the title of his talk interplay of oxidative stress, inflammation, lipid, and cell death signaling pathways, injuries and regeneration in vivo, implications for physiology and pathology. Al, take it away.
Okay, thank you very much, Terry, for the introduction. And as you see from the title, and Terry mentioned that the signaling is complex, but if you go to in vivo models or in humans, the situation is becoming even more complex, and this will be the main message of my presentation today. So this is a scheme, and I will introduce some of these major players which will reoccur during my talk periodically. I might use the mouse. So during the tissue injury, always there is increased formation of reactive oxygen species, which can be generated by various sources like mitochondria, NADPH oxidases, as well as xanthine oxidases, cyclooxygenase, and many other pathways. So when reactive oxygen species is formed, it is almost inevitable that reactive nitrogen species is also formed because NO is always there in cells. And example, nitric oxide and superoxide interacts, forming like reactive nitrogen species peroxynitrate, which degraded and induces a shower of various reactive nitrogen species. So also during inflammatory processes, inducible nitric oxide synthase is always induced, which can generate lots of NO. And then when this happens, so usually this leads to oxidation of various proteins, activation of cell death signaling pathways, lipid peroxidation, and also oxidative DNA damage, which then can lead to the activation of nuclear enzyme poly-D-pyrribose polymerase. This is the most abundant nuclear enzyme. Once which start to repair the DNA, but it uses NAD and it leads to impairment of ATP synthesis for glycolases when overactivated, and it leads to cell necrosis in most of the cases. So this is one of the well-known pathways of cell necrosis. But also through the decrease of NAD levels, recent studies showed in several Cell papers that PARP can play an important role in metabolism because sirtuins, like for example, SIRT1, which is the most characterized one, which may be responsible also for longevity, at least in certain species, is NAD dependent. So if you reduce NAD level, the activity of these enzymes will also decrease. And also PARP, when it ribosylates various nuclear proteins by consuming NAD and leading to ATP depletion, it changes the charge of various transcriptional factors which can move, like for example, active PARP is a co-activator of nuclear factor kappa B, which is one of the key pro-inflammatory pathways, which can regulate expression of inflammatory cytokines, TNF-alpha, chemokines, NAD, pH oxidases, and the inducible nitric oxide synthase. And also when necrosis happens, it will also lead to release of various cellular material which activate the inflammatory response, and these inflammatory cells then produce more nitric oxide and also reactive oxygen and nitrogen species. So ischemic reperfusion injury is one of the predominant pathways of tissue injury during myocardial infarction, stroke, and organ transplantation and vascular surgeries. So the ultimate treatment, when, for example, there is myocardial infarction or stroke and the vessel is occluded, to eliminate this occlusion. But when this happens, reperfusion occurs, which leads to marked surge of reactive oxygen and nitrogen species generation, which lead to the activation of various pro-inflammatory pathways like NF-kappa B, and in endothelial cells, increased expression of adhesion molecules, release of various chemokines which attract then inflammatory cells such as neutrophils, which lead to inflammation and more oxidative stress tissue injury. And this is although this is the treatment of myocardial infarction, is very important to try to limit its damaging effect. So when most of the cases when cells are damaged and die, they can go either to necrosis or apoptotic cell death. So during the necrosis, there is release of various cellular material which can activate danger-associated molecular pattern receptors in inflammatory cells and recruit inflammatory cells and induce pro-inflammatory response. So in case of apoptosis, these apoptotic bodies are undergo phagocytosis and there is no inflammatory reaction, and quite often apoptosis could be a protective way of eliminating damaged cells. So as you see from this scheme, so from necrotic cells, various materials like RNA, DNA fragment, HMG box proteins or ATP, various inflammatory cytokines can signal to inflammatory cells to various receptors and induce pro-inflammatory response. So probably one of the most known is TLR4 receptor, which is the receptor also which is activated by the bacterial endotoxins. So this or some of this can be released from the cells and activate inflammatory cells during the necrosis. So today I will try to illustrate the complexity of inflammation, oxidative stress related processes and some other like lipid signaling using a hepatic ischemic reperfusion model. So in this model, we occlude the blood flow in mice to the 2/3 of the liver, so approximately 70% for 1 hour. And then we remove this clamp and the blood, oxygenated blood returns. And then we studied the events at various time points, like 2 hours, 6 and 24 hours following the injury. So in the liver, the main cellular players are hepatocytes, and then Kupffer cells and sinus endothelial cells. First, when there is injury, the endothelial layer is damaged, and these Kupffer cells are modified macrophages. They are sitting on endothelium, and they can get activated. And later, when these cells are activated, they release chemokines which attract neutrophils. So at the later phase, there is increased neutrophil mediated injury, oxidative stress, and damage. So this is how it looks histologically. So this is a hematoxin stained liver slice. So this is a normal liver hepatocytes, and this is the sinus. So following the injury at 2 hours, you see like edema, loss of blood. But later at 6 hours, you already see like necrotic coagulation, necrosis, and this is very well demarcated at 24 hours. And then AST and ALT, this is liver transaminases, which are released from dying hepatocytes, and these are the markers of acute necrosis. So you see that the peak necrosis occurs 6 hours following injury, and then later the necrotic cell death is changed to apoptotic cell death, where these liver enzymes are going down. So you see also PARP activation. This is a necrotic pathway, which I mentioned at the beginning. So there is a time-dependent increase, and also there is time-dependent increase of apoptotic cell death. But apoptosis occurs only at later time points. So initially there is necrosis, and at later time point there is also apoptosis. So what happens at early stage, you see electron microscopy of livers, and these are mitochondria in control animals, and you see nice electrodense mitochondria with nice structure, this black mitochondria, and following 2 hours of reperfusion, there is marked swelling of this mitochondria, and they becoming white, so they lose the structure and the electron density. So there is mitochondrial injury, and you see also decrease of mitochondrial complex 1 activity, and impaired complex 1 activity can generate reactive oxygen species. And also from isolated mitochondria, there is increase at various carbonylated proteins, and also nitrotyrosine, which is a marker of nitrative stress. So here you see this visualized. So this is HNA ducts, which are stable lipid peroxidation products, and you see time-dependent increase in the ischemic reperfusion. And you also see time-dependent increase of NADPH oxidase 2 isoform, which is mostly in the infiltrating neutrophils. At 2 hours, there are no neutrophils, but later they are coming. And this is visualized, so this is normal liver stain for with lipid peroxidation sensitive marker. You see marked increase of lipid peroxidation 24 hours after reperfusion injury. And so this is a marker of oxidative DNA injury. So this is in the nuclei. You see not much in normal hepatocytes or other cells, and there is increased dark staining in the nuclei, indicating oxidative DNA injury. And then, as I mentioned, nitrotyrosine is a marker of nitrative stress, which is in part coming from peroxynitrite. So with the ELISA, you see time-dependent increase. And so this is the normal sections, no increase. And following ischemic reperfusion 24 hours, there is marked increase. This brown staining, which is localized in hepatocytes, in some of the inflammatory cells, and also in endothelial cells. And here you see that basically this sinus disrupted, and through endothelial layer, these inflammatory cells migrate to the tissue where they mediate injury. So what happens with the inflammatory response? So you see these are various chemokines, MIP1 and MIP2. So there is early increase, which is coming from Kupffer cells, and then attenuation of the chemotaxis, and similarly to the TNF-alpha. But there is time-dependent increase of neutrophil infiltration. So these neutrophils are stained with the myeloperoxidase, which is an enzyme characteristic of neutrophils. So you see practically no neutrophils. You see in the vessels, they start to attach, but they are not in the tissues yet. And they start to migrate to the tissues. And at 24 hours, there is huge neutrophil infiltration. And the neutrophils, they have NADPH oxidase 2 isoform, which can be a significant source of superoxide and can produce various other inflammatory mediators as well. So this is a little bit more details, but still a very simplified scheme what is happening. So basically, during the ischemia by itself, there is mitochondrial dysfunction, complex 1 activity decrease. There is mitochondrial rose generation, and it leads to decreased ATP, which leads to hepatocyte necrosis. So from necrotic hepatocytes, you have release of various these danger-associated molecular patterns like HMG box protein or RNA DNA fragment, which signal to the Kupffer cells. They activate these Kupffer cells, which produce TNF-alpha, activate NOx2 to generate reactive oxygen species. They have inducible nitric oxide synthase, which is a source of nitric oxide. And there is increase of chemokine release. And this leads to the attraction of neutrophils, activation of endothelium, which express adhesion molecules. Then these neutrophils attach to the endothelium, damage endothelial layer, and then they transmigrate and go to the tissues where they produce inflammatory chemokines, cytokines, reactive oxygen and nitrogen species, leading to necrosis or liver dysfunction. And in this later phase, like cyclooxygenase 2 is also a very important player, which Terry already mentioned. It can be a significant source of various prostaglandins, and this is also regulated by nuclear factor kappa B signaling. So here I will show an example how this can be targeted by modulation inflammation and oxidative stress, or by targeted antioxidant to the mitochondria. So marijuana, you probably heard a lot about this cannabis sativa, particularly recently, where it is getting legalized in some states. Is a source of over 400 various compounds. 80 of which are very unique terpenophenols, which resemble different antioxidants. And tetrahydrocannabinol THC is the main ingredient which is responsible for psychoactive effect, but also for some of the anti-inflammatory effect, for psychoactive effect through CB1 receptors in the brain, and anti-inflammatory effect, this is CB2 receptors in inflammatory cells. And initially, when it was discovered, it was thought that this is the only active ingredient in cannabis. But then it turned out that there are lots of other compounds, and one of these is cannabidiol, which was proposed by Julius Axelrod at NIH long time ago, together with tetrahydrocannabinol and also with David Wink, who Terry already mentioned, that these are potent antioxidants. And at that time, nobody believed that cannabidiol is an active ingredient, but now cannabidiol is approved in more than 6 countries to treat multiple sclerosis, a very serious degenerative disorder, and recently it also got FDA approval for the treatment of treatment-resistant childhood epilepsy. So we studied cannabidiol in liver reperfusion injury. So you see here there is increase of liver transaminase marker of liver necrosis 6 hours following injury, and there is marked reduction with cannabidiol. You see necrotic area following ischemic reperfusion, which is largely reduced by cannabidiol. This is neutrophil infiltration following ischemic reperfusion. These are control livers, markedly reduced. And this is the apoptotic cell death. So these are the normal nuclei, the blue. And following ischemic reperfusion, you see this fragmented, this is greenish, brighter nuclei. These are apoptotic cells, which are also decreased. And here I just want to show also the complexity of this various pathways. So these are, this is a chemokine. So you see ischemic perfusion is always red. So these are the controls white, a marked increase and time-dependent decline, which is attenuated by cannabidiol. This is an important adhesion molecule, which can be expressed by endothelial cells and some other cells as well. So also there is marked induction and attenuation by cannabidiol. And this is another chemokine and cytokine TNF-alpha. There is marked increase and attenuation blue with cannabidiol. And also you see compared to the control, marked early activation of NF-kappa pathways in these livers, which is attenuated by cannabidiol, and these are various stress kinases which lead to cell death, mostly apoptotic, so there is early increase, and this is attenuated by cannabidiol. So, and then other players, so you see the H&E, this is a marker of stable lipid peroxidation. There is time-dependent increase with ischemic perfusion and attenuation, and this is COX-2, which Terry already mentioned, that COX-2 inhibitors are very good anti-inflammatory drugs for joint problems, and they do not inhibit cycloxygenase in the gut, so they have less gastrointestinal side effects. However, and COX-2 inhibitors are beneficial also in cancer. But as I will talk later, COX-2 inhibition may not be necessarily always beneficial because some of the COX-2-derived prostaglandins are involved in liver regeneration. So short-term inhibition of COX-2 can be anti-inflammatory like in liver injury, but long-term may prevent the regeneration of the liver. And also COX-2 inhibitors were withdrawn from clinical trials, some of these because of cardiovascular side effects, because some of these prostaglandins may be protective in the cardiovascular system and similarly in the kidney. And now they came back with a warning. So if you read, so if you have cardiovascular or kidney problems, it is not recommended using these drugs because they can increase the chance of myocardial infarction or cardiovascular effects. And for example, in the kidney, COX-2 plays a very important role during the injury. So normally the blood flow in the kidney is maintained by nitric oxide, but if you have kidney injury, COX-2-derived prostaglandins maintain the blood flow. So if you inhibit COX-2, then you basically can kill the kidney. So there is also so-called non-steroid inflammatory drug-induced nephropathy. So if you read carefully these labels of these drugs, you can find this information. So for kidney and cardiovascular problem, OX2 inhibition could be actually not good. So that's why it is very important to try to see a complex view of the events what we target. And so back to the mitochondrial dysfunction, there is decreased complex 1 activity, which is improved by cannabidiol. There is a high throughput source of nitric oxide, which is responsible for generation of reactive nitrogen species when combined with superoxides generated by NADPH oxidases. For example, this is NOx2 isoform. There is marked increase. And nitrotyrosine, as I mentioned, marker of nitrative stress, there is also marked time-dependent increase, and this is attenuated by cannabidiol. And this is nitrotyrosine. So you see this normal livers, no nitrotyrosine, and following ischemic reperfusion, there is attenuation, and the higher magnification shows that it is in endothelial cells, hepatocytes, and infiltrating immune cells as well. So this is some anti-inflammatory effect of abenoids in Kupffer cells. I mentioned that these are very important resident macrophages in the liver. So if you stimulate Kupffer cells with endotoxin, there is marked release of TNF-alpha. And you see also NF-kappa B activation, and this was attenuated by cannabidiol and also the TNF-alpha release of these cells. And in human vascular endothelial cells, if you expose them to TNF-alpha, there is marked expression of through NF-kappa B of adhesion molecules VCAM and ICAM. And this was also attenuated by CBD. This is light blue. And the significance of this, so if you have normal endothelial layer or vessels, so normally inflammatory cells do not attach because there is no increase in adhesion molecules. But once the adhesion molecules expressed, so these greens are inflammatory cells, if you expose them to inflammatory cells, they stick to the surface of endothelium, and this could be attenuated by cannabidiol. So concluding this part, cannabidiol has multiple targets. So one of them is mitochondrial dysfunction and ROS generation, but it also targets inflammatory Kupffer cells and also neutrophil-mediated response. Why it is important? Because normally if you have myocardial infarction or other forms or stroke, so the therapeutic window is usually very short. So usually if patients arrive early, then antioxidant may work well in this phase. But later you have to target the inflammation. So antioxidant, if given in these phases, will not work anymore. So, and we also explored the role of mitochondrial oxygen species generation, and we used various selective antioxidant targeted to the mitochondria, which taken up by the mitochondria and converted to an antioxidant actually, in fact, in the mitochondria. And what we found that targeting this mitochondrial ROS had the same beneficial effects. So mitochondrial ROS was triggering all this inflammatory response and damage. However, when we administered this antioxidant at later phase, they were ineffective. So which means that basically mitochondrial dysfunction and oxidative stress triggers these key inflammatory pathways, but targeting mitochondrial dysfunction is only effective at early stages of ischemic reperfusion. So if somebody with stroke immediately hospitalized, this could be a very promising approach. If not, then this antioxidant will not be effective. But some of the drugs like cannabidiol, which target both inflammation and oxidative stress, they may be still quite effective. So now I will talk a little bit about the role of the endocannabinoid signaling system with oxidative stress. So this is a newly discovered lipid signaling system, and it also has important overlaps with arachidonic acid metabolism as well. So there are two main G protein coupled receptors, CB1 and CB2. So CB1 is expressed primarily in the brain and mediates psychoactive effects of marijuana. The active ingredient, as I mentioned, is THC, which is both CB1 and CB2 agonist. CB2 receptors expressed in the immune cells. And very interestingly, several endogenous ligands called endocannabinoid were identified which activate these receptors, anandamide and arachidonyl glycerol, which surprisingly turned out to be lipids, not peptides. And then this synthesized from membrane lipids, and so anandamide and 2-AG, and they degraded to arachidonic acid. So this is very important. I will talk about that later. Particularly for 2-arachidonyl glycerol, which has approximately 100 times higher tissue levels. So it can be a very significant source of arachidonic acid and prostaglandins during the tissue injury, and these are the endocannabinoid degrading enzymes, FA and MAG-lipase. So if we inhibit this, we can raise the levels of endocannabinoids, but we can also inhibit the hydrolysis to arachidonic acid, as I will show. So as I mentioned, CB1 receptors in the brain, they are responsible for psychoactive effect and they promote metabolic effect leading to obesity and fat storage in peripheral tissues, and also have cardiovascular effect like cardiodepressive effect. But more recently, it also turned out that in addition to this metabolic effect, cannabinoid receptors are also in inflammatory cells, CB1, and CB1 activation in macrophages can induce ROS production and stimulation of pro-inflammatory mediators like TNF-alpha release through MAP kinases. And also CB1 receptor activation in vascular smooth muscle can lead to upregulation of angiotensin II receptor signaling, which through NADPH oxidase is also significant source of reactive oxygen species. So in contrast, CB2 receptors are primarily in inflammatory cells, particularly in certain lymphocytes and also macrophages. And stimulation of these receptors is in general anti-inflammatory. So what happens if we stimulate these receptors? It decreases chemotaxis of these inflammatory cells, endothelial cell activation. Endothelial cells also have CB2 receptors, and then migration and transmigration and activation of these cells. And this has been shown to be beneficial in various models of ischemic reperfusion injury. And where are these receptors in the liver? So this is just, you see the spleen, this is flow cytometry. So this is without the CB2 antibody, the black and blue is red. So you see that B cells, macrophages has loss of CB2. In the liver, in hepatocytes, there is no CB2. And there is CB2 in Kupffer cells, which are macrophages, in neutrophils and endothelial cells as well. But there is no CB2 in hepatocytes. This is just keeping in your mind. And we explored the role of CB2 signaling in the liver. We used a synthetic CB2 receptor agonist, which target the anti-inflammatory CB2 receptor. So you see liver enzyme marker of liver necrosis increased. This was attenuated by CB2 receptor agonists, and there was bigger injury in CB2 knockout mice, suggesting that endogenous cannabinoid signaling through CB2 may be protected. And similar effects were seen on various serum and liver inflammatory chemokine cytokine that ischemic perfusion increased the injury, and this was attenuated by CB2 receptor agonists. So you see marked necrosis during ischemic perfusion. This is more focal, less necrosis with CB2 agonists, and there is much bigger necrosis in CB2 knockout mice. So this is neutrophil infiltration reduced, and more neutrophils in CB2 knockout mice following ischemic perfusion injury. And these are endothelial cells, which following stimulation express adhesion molecules. There is big increase, and this was attenuated by CB2 receptor agonists and reversed by the antagonist, suggesting that it is CB2 mediated. And when we take this endothelial layer, stimulate with TNF-alpha and put inflammatory cells, neutrophils, they stick more to the activated endothelium, and this was also CB2 receptor dependent. And here we developed a new CB2 receptor agonist, which is now in clinical development for kidney ischemic perfusion, kidney disease, and diabetic complications. So you see a marked attenuation of necrosis by this drug, and also marked attenuation of inflammatory response, and also attenuation of inflammatory response in the macrophages in Kupffer cells. So the more interesting thing was what happening with the endocannabinoids. What is the role of these endogenous lipid mediators and how they interact with the oxidative stress? So as I mentioned, the two main endocannabinoids are anandamide and 2-AG. So you see marked increase following ischemic perfusion in the liver, like 20-fold and here 4-5-fold. And this was originated not only from inflammatory cells, but also from hepatocytes. And there was correlation of liver injury measured by this liver transaminases with the levels of endocannabinoids in the liver. So the more endocannabinoids were released, the bigger the injury was. And we also hypothesized that most likely reactive oxygen species leads to the generation of these lipid metabolites, because during the ischemia by itself there was very little increase, and this happened during the reperfusion where this massive surge of reactive oxygen species happened. And then we exposed hepatocytes to various pro-inflammatory mediators or oxidants like peroxynitrite, hydrogen peroxide. There was marked increase of these endocannabinoids, suggesting that ROS leads to increased production of these endocannabinoids. And this is just keep in mind that these are degraded by two main endocannabinoid metabolizing enzymes, arachidonic acid. And arachidonic acid is a significant source of prostaglandins in the liver. So here we wanted to know what happens if we inhibit the endocannabinoid degradation, this MAG lipase enzyme, which raises the 2-AG levels in the liver. So you will have more 2-AG, and we found that inhibition of this enzyme or genetic deletion was associated with marked reduction of injury. So this is necrosis following ischemic perfusion. This is a normal livers, and marked attenuation. And there was marked reduction of inflammatory response. So these are infiltrating neutrophils, and there was a reduction. When we explored the mechanism, we hypothesized first that it is mainly mediated through CB2 receptors, this protective effect on inflammatory cells. as I mentioned, that pharmacological stimulation of CB2 is anti-inflammatory and can decrease the growth generation in inflammatory cells. However, there was a very interesting paper of Ben Kravat in Science in around 2012, where they showed that this enzyme is primarily one of the very important hydrolytic pathways of 2-AG to arachidonic acid in the brain and as well as in the liver. And they showed in this science paper in the brain, and they inhibited this enzyme, there was a marked protection against endotoxin-induced brain inflammation, which was associated with the decrease of various prostaglandins. So it behaved like a sort of COX-2 inhibition because basically it reduced the precursor for COX-2-derived metabolites. And we studied the same in the liver. So you see here in normal livers, following inhibition of this enzyme, there is increase of 2-AG, and there is increase during the ischemia reperfusion. The other endocannabinoids is not modified, and there is marked increase of arachidonic acid, suggesting that it is coming from 2-AG. And also there was increase of various prostaglandins, COX-2-derived prostaglandins in reperfusion injury, and this was attenuated by inhibition of 2-AG metabolism. So how this comes together? So during the acute liver injury, there is cell necrosis in hepatocytes, but hepatocytes have no CB2 receptors. So when we block monoacylglycerolipase, we raise 2-AG, which can release from hepatocytes. It can decrease the endothelial cell activation through CB2 and also Kupffer cell activation and neutrophil-mediated injury. But on the other hand, we also decrease the arachidonic acid formation and production of COX-2-derived prostaglandins. So inhibition of this enzyme in this acute injury models in the liver and in the brain behaves like a combination of a CB2 inhibitor and the COX-2 inhibitors. But as I mentioned, that inhibition of COX-2 during the kidney injury or heart injury, long-term inhibition may be problematic because some of these prostaglandins are protective. And for example, prostaglandin E2, which are showed in the previous slides, plays very important role also in the liver regeneration. So if you have following liver transplantation, you limit the ischemic injury by this approach of COX-2 inhibitors. This is acceptable. But if you want to use them very chronically, in chronic liver disease, this may be problematic because prostaglandins play a very important role in tissue regeneration and liver regenerate very fast. For example, in mice, if you take out 2/3 of the liver, it regrows in 1 week, and most of it regrows in 2-3 days. So liver has, unfortunately, the regenerative capacity in humans is less, but it's still very significant. So it is very important not to inhibit this process. And this is an example for the same that in other tissues or organs, like this is a acute heart failure model. And I will just show what happens if we inhibit one of these endocannabinoid degrading enzyme, which was protective in acute liver injury. So in this model, we use doxorubicin, which is a very widely used chemotherapeutic drug, which is known to induce mitochondrial oxidative stress. And unfortunately, it can induce heart failure, which sometimes occurs only months or years after the treatment with this chemotherapeutic drug, and this is very widely used, like in breast cancer, in various other tumors. So this is a mouse model of acute doxorubicin-induced heart failure. So you see that 5 days after administration of high dose, mice start to die. But if we inhibit this endocannabinoid degradation, practically all mice die. And in the myocardium, with doxorubicin-treated mice, there is increase of cell death, apoptosis, and there is much greater increase when you inhibit the endocannabinoid metabolism, and similarly reactive oxygen species footprints like lipid peroxidation product, nitrotyrosine is the same. And normally this is an acute reactive oxygen species mediated necrosis in the heart, no inflammatory cells. But if you inhibit the endocannabinoid metabolism, there is also increased inflammation in the myocardium. So this is again showing the complexity that you target something in one tissue or during certain part of the disease, it may be good. But if you target chronically, it may be actually detrimental or induce the opposite effect. So very briefly, endocannabinoid system is activated by reactive oxygen nitrogen species during the tissue injury, and through cannabinoid receptors, it can induce the protective effect for CB2 or detrimental for CB1, and these both receptors are involved in the reactive oxygen nitrogen production on inflammatory and some other cells. But some of this effect can be mediated through pathways unrelated to CB2, CB1 receptor, through modulation of the arachidonic acid cascade and COX-derived metabolites. And selective CB2 agonists are anti-inflammatory and now developed in treatment of kidney reperfusion, diabetic complication, and periphery restricted CB1 antagonists. They are very beneficial in obesity, diabetes, diabetic complications, and also more and more studies show that this signaling also importantly overlaps with the reactive oxygen species generation. So here I just want to show a brief example when reactive oxygen species in fact can be beneficial and can play a very important role in tissue regeneration. So this is an interesting, relatively recent paper in Nature Cell Biology. So they used this Xenopus tropicalis tadpole, so which is a larva form of an amphibian frog, because it has very good regenerative capacity of the tail. So if they cut the tail, it regrows within a couple of days. And actually, strikingly, these processes are very similar, like in regenerating liver. For example, I mentioned that in mice, liver regrows within 1 week. So this tail also regrows within 1 week, and the major processes of regeneration occurs during the first 2-3 days. And this is also very similar here. So what you will see here now, that here this is an early phase. They just go up to probably 2 days. So this is when the tail will regrow. Try to activate it. And the lower part is when they express the construct, which is detecting the changes in hydrogen peroxide. So the more, if the scale moves to the white, red and white, then it means that you have more production of reactive oxygen species like hydrogen peroxide. And here you will also see windows where they just trace the inflammatory cells. So I will, hopefully it will work. So you see that the tails start to regrow up. This is just a normal microscopy, and there is increase of the white and red, which is the reactive oxygen. And these are the migration of inflammatory cells in the lower trace the AR trace. So it really looks really cool. And you have more ROS production. And importantly, the ROS signal comes first before this inflammatory cells start to go and facilitate the growth of this tail, which means that it is occurring not in inflammatory cells, and the inflammatory cells in this case not the predominant sources of ROS. So here is this species, how it looks. So this is when they cut the tail, and this is just regrowth in 7 days. So we went up with the video approximately to this, I think, 2 days. And you see here this construct. So if they expose to certain certain wavelength, you see the red and white is the top of the scale. This shows marked increase of reactive oxygen species as it starts from 6 hours, 1 day, and then later it decreases. When they inhibited, they used 2 inhibitors of NADPH oxidases. One is just a flavoprotein inhibitor, DPI. The other is apocinin, which inhibits the assembly of NADPH oxidases. Like Terry mentioned, you will learn about this ROS producing enzymes later. There was reduction of ROS and reduction of the tail regeneration. So you see here, this is just DMS, so this is early, this is after 7 days, tail regrowth, and it was markedly inhibited by inhibition of the NADPH oxidase. But when they used the global antioxidant, this MCI, which is also in clinical trials, it reduced a little bit, much less the ROS production was compared to this Y than the other 2 approaches. But interestingly, it did not inhibit that much. It also impaired the tail growth, but later they also did an experiment when they inhibited this process during the first 3 days, and they washed out. And in this case, if they inhibited NADPH oxidase, the regeneration would not occur anymore, so it was sort of irreversible. They would not regrow. But if they just washed out this global antioxidant or reactive oxygen species scavenger, this process could be reversed, and to a certain extent, tail almost completely was regrown. And then they linked this to very important signaling events which are involved in the tissue regeneration. So here, this is an example where reactive oxygen species signaling is very important in a physiological, or you can say a pathological process, and the same exactly happening like during the liver regeneration when reactive oxygen species can be very important signaling molecule, and if we drastically inhibit the ROS, then we also see impaired liver regeneration. And more interestingly, if you target specific forms of ROS, like ROS generation enzyme or in the mitochondria, you may have completely distinct effects. And most of the antioxidants in clinical trials are failed, and probably one of the reasons that they do not specifically target the forms of ROS, or they target at wrong time, because the timing is also very critical, because like you see here, ROS initiates something. If you target at right time, it may have a profound effect, like in ischemic reperfusion. If you target later, it can have the opposite. Or the same with COX-2 inhibitors, like they are very good in certain sort of kinds of cancer, in arthritis, and COX-2 inhibitors, they do not cause gastrointestinal side effect. But if you have kidney disease, it can be a problematic in the kidney, because some of these prostaglandins in damaged kidney maintain the normal blood flow, and the same in the heart. And the same in the liver, like reduction of COX-derived prostaglandins acutely beneficial, but chronically it can impair the regeneration. So basically, the conclusion is, again, that the interactions of this reactive oxygen species with inflammation and other, like lipid signaling pathways, are very complex. It is even more complex than in vitro, and it is very important to consider the pathology and knowledge of the pathology like this is extremely important, and various stages when and what exactly happens, because targeting certain processes at the right time can be very beneficial, but the same approach when used at wrong time may induce a problem and may be detrimental. So finally, thank you very much for our scientific director and people in my lab and collaborators, David Wing and Terry, who have been involved in some of these studies. Thank you for your attention. Yes, but you know the problems that, I mean, the selective CB2 receptor agonists are developers, but with the marijuana by itself is the problem that you have THC there, which is a non-selective CB1 and CB2 receptor agonist. And nobody wants to go into the psychoactive effect because, you know, this is a problem. And then CB1 receptor activation is not good in metabolism. It can promote also growth generation, diabetes development. So probably why marijuana is not causing bigger problems, because it has lots of other antioxidants like cannabidiol, which counterbalance the detrimental effect of CB1 activation by THC, and also the CB2 effect of THC can also counterbalance this. And also the lucky case that THC is a relatively weak CB1 receptor agonist. But now there are the designer drugs, which you have probably heard, like Spice. It is very popular. It is a mixture of various synthetic CB1 agonists. You can buy most of this even online, because there are only 5 which are prohibited. But if there are others you can buy, and some of these contain drugs which 100,000 times more potent than THC. They are not regulated. And now there are some clinical studies, like in young children from CDC, showing that use of this drug is associated with acute kidney failure, chronic kidney disease. So kids who are smoking this couple of times, they end up on dialysis. Who smoke them chronically have bigger problems. There are reported cases of myocardial infarction in children using these substances, because CB1 is bad for your cardiovascular system. But certainly there are very good ingredients in the, I mean, like cannabidiol. Now it is already approved an extract which contains very, very low amount of THC. It has cannabidiol and some of the other these terpenes approved in like 7, 8 European countries. In Canada now it is pending FDA approval in multiple sclerosis, which is a chronic debilitating neurodegenerative disease. And now it got approval of cannabidiol alone in resistant cases of childhood epilepsy. Basically, nothing is effective. So from 2 different companies was approved like a few months ago, and it is working very well. And for example, in childhood epilepsy, when you have continuously these epileptic seizures, you have continuous excitotoxicity in the brain, which also leads to massive growth generation and injury. And if you don't stop this, I mean, eventually you will have very serious brain damage and this kid's development is completely delayed, and they have also mental issues later. So, but again, when it was proposed by Julius Axelrod, who, by the way, got Nobel Prize for something else, nobody believed that it is an active constituent in marijuana, and that it is an antioxidant that was shown in PNAS paper by Julius Axelrod and David Wink, and even was called neuroprotective antioxidant from marijuana, and in fact now it is used in sclerosis multiplex. So it is interesting. Thank you.